So hello class. So let's see how these pathogens can be eliminated. 1. By binding onto a pathogen, by binding onto this, what's the name? By binding onto pattern recognition receptors, a pathogen can be eliminated. We have lysozymes, complement system, C-reactive proteins. So binding of pattern recognition receptors on the host. To an example of these pattern recognition receptors are located on the lysosomes, the complement system, C-reactive protein of the host. So these are the chemicals that will recognize this antigen and bind onto them, and it will result in direct lysis of pathogen. Lysis means that the pathogens will be broken down, or their cell membrane will rupture, their cell membrane will break apart and drain its content. So this is the cell nucleus undergoes rupture or lysis. So you can see it is broken, and the content will come out like this. It will pour it out. Okay, so direct lysis. We have opsonization followed by phagocytosis. Opsonization means coating. So when we see the microorganism, if this is a virus, this is a virus, then when the immune system recognizes it, it will coat it with antibodies. It will coat it. This is an antibody, so it will coat it with antibodies. So after coating, then after coating, then the macrophage is a phagocyte. Dendritic cell is located in the skin. Dendritic cell and neutrophil, which is also called granulocyte, and neutrophil will come and feed or phagocytose this. Phagocytosis is like this. If this is a macrophage, this is a macrophage, then this is an infection. The macrophage will come, it will open its cell membrane around this, like this. Then it will form a food vacuole. It will engulf it. So now these two viral particles will be inside. Then it will attach it to what? A lysosome. L for lysosome. Then the lysosome will release its content and kill this organism. So this process of wrapping the cell membrane around the organism, the disease-causing organism or pathogen, and producing lysozyme, to kill the organism in the vacuole. The process is called phagocytosis. That is cell eating. It is eating up a cell. But if you say pinocytosis, if it is pinocytosis, it means pinocytosis is what? Cell drinking. Pinocytosis is what? cell drinking. It means this organism, this virus, instead of it being a virus, it is a chemical or a liquid. A liquid. It is a liquid. So the cell will now drink the liquid. It is no longer cells, it's liquid. So the cell will wrap its cell membrane around the liquid here and engulf the liquid and destroy it. That is what we call cell drinking, pinocytosis. And if it is cells, then the macrophage, which is antigen presenting or phagocyte, will wrap around it and produce lysozyme to feed on it. And that is what we call cell eating. Cell eating. Cell eating. The cell is eating the virus. Okay, let's continue. So by direct lysis, so opsonization is coating. So when we coat the surface with antibodies, or we coat the surface of a virus with complement, and the complement we use for coating is C3b. C3b. So C3b complement is a protein. It will coat the outside of the cell, of the plasma membrane of the organism. This C3b is produced by the body, by the human host. So it will come and coat the cell membrane of this organism. So when the immune system recognizes the C3b, which has coated this virus, then the immune system will be awakened. The macrophage will be produced to come and feed on this virus. The macrophage will be produced to come and feed on it because it has been coated. It's like this. If you see a lady who has dressed to kill, you know that the lady is in for action. She wants a man to fall. If you see a lady who has dressed to kill, it means that the lady is poised for action. She is tempting somebody. So the men who don't have self-control will now go in and sleep with her and give her HIV. So that is what coating. The lady coated herself with lipsticks. She even wore dresses that expose her body. So we say that that lady has coated herself with makeup. So she is liable or vulnerable to be destroyed by dangerous, gravinous, wicked men. So that is coating. So we have direct phagocytosis, or that's the cell eating, via or through cell associated pattern recognition receptors. That's what I explained. Enhancement of phagocytic cell function. That's what I explained. Macrophage eating a cell, or drinking a cell, drinking chemical from a cell. Production of cytokines and chemokines, we'll get there. Then differentiation of effector cells to more active states. So the innate immune system has these components. We have the epidermis and the dermis. The epidermis is just the outside of your skin. The outermost layer of your skin is the epidermis, or the outermost layer of your body. It comprises of what? Stratified squamous. Stratified squamous. The epidermis comprises of what? Stratified. Stratified squamous You will do this in histopathology, epithelium or epithelial cells. stratified squamous epithelial cells. So that's the outermost. Some of them are keratinized, meaning that they have keratin, that's for the hair. Keratinized, others are not. Those who are located inside the body are non-keratinized. Outside the skin, they are keratinized. So simple stratified squamous epithelial. But the skin is liable for wear and tear. So let's go. So we have the first, the skin is a mechanical barrier, a physical barrier. It will prevent the microorganisms from entering the human host. So it has tight cells as barrier to pathogens. Continual division of cells will slough off the pathogens. So if pathogens are on the skin, when you even bath, they will be washed away. When your cells of the skin divide through mitosis, they will be washed away, or they will be broken or destroyed, or they will be released. So they will be sloughed off the skin. Dendritic cells act as phagocytes, like I explained phagocytosis. Dendritic cells are cells that are antigen presenting cells found in tissues, and it is also a type of phagocyte. So skin epithelial comprised, so it's also a phagocyte. So let's go. So we have the dendritic cell. We will come to it later when we do antigen presentation. So the dermis of the skin, after the epidermis, we go deeper into the dermis of the skin. So if this is the skin, hey, sorry, my drawing is bad. If this is the skin, this first layer is the epithelium. And the next is endo. This layer is endothelium. We have the epithelium and the endothelium. So the dermis comprises, it has collagen. So the epithelium, so this one is epidermis, I was going to write, this is epidermis, and this one is not an epithelium. Your lecturer is sleeping. It's not easy to do recording. My young ones, it's not easy, just sleeping. So this one is what? Dermis. So for the epidermis is the outermost layer of the skin, followed by, when you do anatomy, instead of we saying that followed by the dermis, we say that followed by the subcutaneous before you go to the dermis. But this is for basic. So we have the dermis has collagen, and the collagen gives strength and pliability or flexibility to the skin and prevents penetration of the skin. We also have the dermal cells which secrete antimicrobial peptides. So the cells of the skin, the dermis, produce defensins, which are antimicrobial peptides. So you see the skin has a whole lot of mechanisms to ward off infections. So we have sweat glands also secrete sweat, and the sweat contains antimicrobial peptides called dermicidins. The sweat contains lysozymes that destroy bacterial cell wall. The sweat contains salts that draws water osmotically from cells. So the dermis has what? Sebaceous gland, which secretes a chemical called sebum. And this sebum keeps the skin flexible and less sensitive to breaking and tearing. The sebaceous gland also contains uh, the dermis also contains fatty acid. No, the 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 sebum contains fatty acids, which has what a lower pH, which is toxic to bacteria. We know that acid can kill bacteria, so the secretion, which is sebum from the sebaceous gland, contains fatty acids, which is an acid. So it lowers the pH to approximately 5. From the normal pH is 7. So above 7 is basic, below 7 is acidic. So this is an acidic pH which will fight the bacteria. So we now look at mucous membranes. So mucous membranes are what? Coverings of the lumen of the respiratory, digestive, reproductive, and the urinary tract, and they are opened, they lead the openings of the external environment. So all these sections of the body, these lumens and the respiratory, starts from the nose and goes back to the nose, digestive from the mouth to the anus, reproductive, that's the vagina. They are all open to the external environment, and they have what? Mucosal mucous membranes, mucous membranes in their endothelium or in their luminal space. So the mucous membrane prevents the organism, kills the organism, because the organism, when they fall into that slimy mucous membranes or into that slimy mucus produced by the cells of the mucosa, then they will swim inside and they cannot wake up because it's too slimy and they will die. So we also have layers. We have the superficial epithelium or thin layer and deep connective tissue, mechanical and nutritive support. So we also have tight epithelial cells prevent pathogens entry. So the first one is the mucous membrane. Second one, the layers of the skin have superficial epithelium, like what I explained, and the deep connective tissue, which provides mechanical and nutritive support. We also have tight epithelial cells, prevents pathogen entry. So the skin has tight epithelial cells. So these epithelial cells, they are tight. They are stratified squamous, and they are tight. There is no junction. There is no junction between them. If these are the cells, there is no gap between the cells. Their cell-to-cell adhesions are very tight. So there is no gap here, no gap here, no gap or hole. There is no hole here. They have tight junctions. So we also say that cytokinesis is the division of the cytoplasm, or when a cell divides into two, the cytoplasm divides into two. That is what we call cytokinesis. The cytokinesis of stem cells continually replace cells and carries attached microorganisms away. Okay, let's look at dendritic cells. Dendritic cells phagocytose invaders. Sticky mucus by epithelial cells traps pathogens. So I've said the sticky mucus already. Dendritic cells are located in the skin. Dendritic cells are located in tissues. They are found in tissues, and they are phagocytes. They are also antigen presenting cells. Chemical substances in mucus. So the mucus produces lactoferrin, lactoperoxidase, and lysozyme. So mucus, that slimy thing that you have been spitting mucus, you blow your nose and phlegms and those things, your vagina juice and the mucosa that makes your vagina so slippery. contains lactoferrin, lactoperoxidase, and lysozyme. Mucociliary coverings of epithelial cells of the respiratory tract moves trapped, so the mucus also has what? Cilia. Cilia are what? Hairs. Small hairs. So if this is the epithelium, we have small hairs on the mucous epithelium, small hairs. So it provides what? A brush border. It serves as a brush border which sweeps away microorganisms. So microbial antagonism. We have the microbial flora prevents establishment of pathogen. I earlier explained this. The normal microflora, they don't kill the pathogen, but they will prevent the pathogen from growing by competing with it for water, food, sunlight, energy, and everything, and even for space. Competition for colonization sites and nutrients, like what I've said. Production of substances. They also produce, the normal microbial flora produces bacteriocins that kills bacteria. Examples are intestinal microbiota. This microbiota or normal microflora they are located in the intestines. Improves health. The normal microbial flora produces biotin, which is a coenzyme, vitamin B5, they are all coenzymes, folic acid, purines and pyrimidines, that's nitrogenous bases. Then vitamin K, gluten, black gluten agent. So we have so the normal microbial flora, these are the substances they produce. Then normal microbial flora decrease in microflora may lead to overgrowth of opportunistic pathogens, and this is referred to as superinfection. Okay, so let's look at the saliva. The saliva contains lysozymes and antimicrobial peptides. Saliva is so powerful, so all the secretions in our body are harmful to organisms. That's why when you take in food, before you even get to the stomach, the organisms that it will meet, that will accompany the food, some will be killed in the saliva, some in the esophagus, some in the stomach acid, some in the duodenum, the pancreatic juice, and some in the small intestine and large intestine. So in the stomach, we have the stomach acid. The bowel also has bowel acid. We have intestinal secretions. They digest and inhibit microorganisms. Gastroferrin, this one sequester. When we say sequester, it holds. Sequester means what? To hold something, or to grab, something. So the gastroferrin sequesters absorb iron, making it unavailable for microbes. So the microbes, they feed on iron, and they become so strong and they destroy the body. But gastroferrin prevents it. It keeps the iron from them, hides the iron from them. The constant movement of the esophagus, of the gastrointestinal tract, the peristalsis. You all know peristalsis. Movement of, if this is the esophagus or the stomach. You can see when there is a bolus of food here, this one will move, the esophagus will move like this. Because it will apply pressure here, pressure is here, so it will contract here, then here will expand for the bolus to move. When the bolus gets here, it will also contract here, so that the intestine over here will squeeze inside, so that there will be a big hole here, then the bolus will travel here too. It will follow like this to get to the stomach. The stomach will also do the same. So Peristalsis moves GIT, your gastrointestinal tract content, constantly. By this action of move peristalsis, it eliminates microorganisms. Then defecation, if you are poisoned by food or through food, when you eat food infected with microorganisms, you realize that you start going to toilet or you start vomiting. Anytime you produce the poo or the excreta or the toilet, there will be a lot of microorganisms there. So that's what they say when you are running diarrhea, just take in minerals. Just take in this electrolytes, because when you take in more of the electrolytes, it will wash off. It will replace the salt that you have lost or the minerals that you have lost, because the more you go to the toilet, the organisms will be coming out. The more you go to the toilet, the organisms are coming out. So you end up going to toilet and removing all the organisms. But if you don't replace the salt by taking if you don't replace the minerals by taking in ORS or a lot of fluids, you end up becoming comatose or getting into coma. So vomiting is also the same. You vomit. When you vomit, whatever vomit that comes out contains the organisms. The more you vomit, the more the organisms are released. But if it becomes too much, you will faint because you will be dehydrated. Okay, so let's look at the urine. So all these are innate immunity or first line. So the urine contains lysozymes. Acidity of the urine inhibits microorganisms, washes microbes from ureter and urethra. Vaginal secretions, that is, the ladies are blessed because the vagina is acidic. So because it's acidic, there is no infection in the vagina, because the acid environment will kill the organisms. So every woman's vagina is sterile because the acid content makes it impossible for any disease-causing organism to dwell there. Prostate secretions. The prostate in men, grown-up men, produce prostate secretions which have properties of binding to iron and preventing microorganisms from feeding on the iron. The menstrual flow cleanses the womb and the vagina. So when you are having menses, it is also clearing your womb and your vagina, and it's protecting you from organisms. When cardiovascular system, when the blood flows, it removes microorganisms. Coagulation or clotting prevents entrance of pathogens, because when you have a cut or a wound, whenever you have an injury or you have a cut, you realize that there will be fibrin formation. So the coagulation and the fibrin formation, the reason why there is fibrin formation is to prevent the cut site and the injured part of your body from being exposed to the external environment. That is why there should be something to cover it, and that cover is what you call what? The fibrin. And the fibrin is gotten from what? Fibrinogen. And the fibrinogen, fibrinogen. This is what we call produces fibrin, and the fibrin is called blood clots. Blood clots. Okay. So transferrin also binds. Transferrin is an ion transporter. So when it binds to iron, it makes it unavailable for microorganisms. For microorganisms, they love iron. So the innate. Okay, let's look at the second line. The second line of immunity, of immune response, comprises of cells, and the cells are phagocytes. It also comprises of antimicrobial chemicals like proteins, like peptides, complement, and interferon. We'll be looking at them. We also have cellular processes like fever. If you have high temperature, it is called fever. And that high temperature, the organisms, they don't like that high temperature, so they will die. So that's why we say you have fever. We have inflammation. We'll be looking at it. So defense components of blood. The blood is made up of what? Cells suspended in plasma. So the blood is plasma. That's the yellowish part of the blood. And the blood, the yellowish part of the blood, if you put it in a plain tube or serum separator tube, it will clot. So after clotting, when you pour out the yellow liquid that is left, that is what we call serum. But if you put it in EDTA tube and it does not clotted, then it will have the clotting factors. So that's the difference between blood plasma and blood serum. You have to find the difference. It's very important. You'll be meeting it in the exams. Okay, so plasma proteins. Plasma proteins, you know, in the blood you have proteins that are called plasma proteins. Examples are what? Example is the albumin, globulin, albumin, globulin, and total protein. They are plasma proteins. So we have plasma proteins. They play a role in inflammation, blood clot, that's the fibrinogen, and infection. Plasma proteins are ion binding. Some of them bind to iron. We call them transferrin. Some of them also act as the complement. And antibodies are also plasma proteins. So antibodies, I forgot to add the antibodies. They are also plasma proteins. An antibody is a protein, the plasma protein. We have complement. It's also a protein. Complement system. They also comprise of proteins. Okay, so let's go to the next. Okay, so defensive blood cells. Erythrocytes, platelets, leukocytes are the formed elements in plasma. The leukocytes, so when we say formed element, that's the solid part of the blood. Solid part of the blood, they are the erythrocytes, or red blood cells, platelets, and the white blood cells, which are called leukocytes. And the leukocytes are made up of granulocytes and agranulocytes. So let's see the example. Granulocytes, examples are what? Basophils. And the basophils are released during inflammatory. The basophils are non-phagocytic. They don't engulf they don't engulf a pathogen. They don't circle around the pathogen and form a vacuole or a vesicle and produce lysosome and kill the organ. They don't swallow the pathogen. That's what I want to say. But they release inflammatory cytokines that fight the disease-causing organism. Eosinophils are phagocytic. They engulf the organism and eat it up. Eosinophils, they mediate the white blood cells that mediate parasitic infections. So this is how eosinophils look like. They have a spectacle nucleus which is like a spectacle. This is an eosinophil. So this is their nucleus. Its nucleus. Eosinophil. And this is the cytoplasm. eosinophil. This is the cytoplasm. Cytoplasm is here, nucleus is here. The nucleus is bilobed. It's a spectacle shape. So we also have the neutrophils. Neutrophil has what? It's a polymorphonuclear. It has what? Multilobar nucleus. The nucleus is lobed. It can be 3 or more. It can be plenty like this. One. The neutrophil has lobes. We call it the granulocyte because the cytoplasm produces granules. So neutrophils are phagocytic. Bacteria, they mediate bacterial infection, and they produce what? Primary eosinophilic and secondary specific granules. Like I said, that's why they are called granulocytes. Anytime you see the full black count results and you see granulocyte, that's neutrophils. So we have 8 granulocytes, and the 8 granulocytes are the monocytes. They mature. Anytime you hear monocyte, know that they will mature to become macrophage and dendritic cells. The lymphocytes are, like I told you in the anatomy, it comprises of the B and the T lymphocytes, and they mediate adaptive response. Then the natural killer cells, they are also lymphocytes, but they mediate innate immunity. They only fight and kill viral infected cells. The macrophage name depends on the location. We have the wandering macrophage, a macrophage that is roaming about in the tissue or all over the body. We have alveolar macrophage, that is macrophage located in the lungs. And the microglia macrophage is contained in the central nervous system. So this is what happens. Let's see. So we have the erythroid stem cell here producing the erythrocytes. Whenever you hear stem cell, blood stem cell, that is what the ancestor cell that produce all these cells. So you can see the erythrocyte. This is the red blood cell. Under the myeloid progenitor cells, they have the platelets, the basophil, the neutrophil, the eosinophil, and the monocytes. I may ask you, what are the myeloid progenitor cells? You should know. Then the lymphoid stem cells, they are the lymphocytes, like the T lymphocyte, B lymphocyte, then the natural killer cells. So the second line of immunity, the processes involved are what? Phagocytosis, that is cell eating. Non-phagocytic killing by leukocytes, that is the leukocytes, the eosinophils, natural killer cells, and the neutrophils. They kill the organism without eating the organism up. They just produce chemicals and kill it. The chemicals, some of them are cytokines. Then the non-specific chemical defenses. We use the toll-like receptor, the nod-like receptor, interferon complement to provide the non-specific. Non-specific means that you cannot identify the type of microorganism you are killing. You are just killing anyone that comes without identifying whether this is a virus or this is a bacteria. We also have inflammation mediating the second line of defense. We also have fever, high temperature. When your temperature is high, it means that the heat that is produced will kill the microorganisms. So phagocytosis. Phagocytic WBCs are called phagocytes. I've explained phagocytosis already. The process of engulfing foreign material by phagocytes is termed phagocytosis. Phagocytes include neutrophils, eosinophils, macrophages. I've already explained. Professional phagocytes are the neutrophils and the macrophages. I may ask you, what are professional phagocytes? Then there are also neutrophils and macrophages. They are also called antigen presenting cells. Antigen APC. Antigen, I'll teach this one in the next lecture. Presenting cells. Okay, so let's look at phagocytosis. Phagocytosis involves chemotaxis, that is directed migration of phagocytes caused by chemotactic agents. Okay, attachment. Phagocyte attached to object, enhanced by opsonization. So before you phagocyte, there should be what? Lipstick. There should be what? Polishing. The woman should polish her face before she can attract the bad boy. And so coating with complement, coating with antibodies, so that they can be recognized by what? The phagocytes, so that the phagocytes will engulf them. We also have ingestion. Ingestion means eating. So the pseudopodia surrounds the object like I explained earlier on, and is taken into the cell in a membrane-bound phagosome. Then digestion takes place. So let me explain. So this is it. The microbe or the disease-causing particle is here, number one. This is the cell membrane. The cell membrane opens up. There is a pseudopodia here. It opens up. Then when it comes to terrain, this opening now forms a vacuole. Lysosome attaches to the vacuole to become phagolysosome. Then there is digestion, partial digestion of the microbe. Then residual bodies, so indigestible materials, are released outside. Okay, so let's look at non-phagocytic killing. That is killing of pathogenic invaders without phagocytosis. So we want to kill them by secreting cytoplasmic granules. So eosinophils secrete antimicrobial chemicals or extracellular toxins and attack parasite helminths. So eosinophils, they are noted, they are the white blood cells which have spectacle-like nucleus, and they mediate or secrete antimicrobial chemicals or extracellular toxins that attack parasites or parasitic helminths. Or parasitic helminths, whenever you hear helminths, helminths is the same as worms. Worms. So then we have the lipopolysaccharides of gram-negative bacteria. They also trigger eosinophil release of mitochondrial distin. Then there is conjugation of the DNA, the DNA of the eosinophil with the eosinophil to form barrier, and this barrier binds and kills the bacteria. So let's look at the non-phagocytic killing. So for the non-phagocytic killing, like I said, killing by natural killer lymphocytes. So we also have the natural killer cells, and they are the third class of cells under the lymphoid progenitor cells. So we have the natural killer cells of the innate immunity, and they secrete toxins against viral infected cells and tumor cells. So when you say tumor is what? This tumor is what? Cancer cells, cancer cells. So natural killer cells secrete toxins or poisons against viral infected cells and cancer. So they only fight viruses and cancer cells. So they do this by employing two ways. One is by deaf receptor pathway, that's they elicit their signals through an antigen receptor pathway, and they also produce granzymes which kill by proteolysis of proteins with target cells. So these granzymes are chemicals that kills microbes. Then we also have, under the non-phagocytic killing, the killing by neutrophils. So when we say non-phagocytic, that means that this phagocytosis mechanism will not occur when they are eliciting their combat or their fight. So killing by neutrophils. So we have two ways by which neutrophils destroy invading pathogens. One, neutrophils produce free radicals or superoxide radical. The superoxide radical is like this. SO8. Superoxide is like this. 2O to is it superoxide? Maybe you can go and check. Maybe is it O3 is ozone. So maybe this one is the correct. Okay. So they produce free radicals, and they also produce extracellular trap nets, what we call cellular suicide, meaning that it will degrade, the neutrophil will degrade its nucleus and cytoplasm and die with the disease causing organism. So we call it cellular suicide. You say the neutrophil will say, my brother, I am Osama bin Laden. I want to kill you, but I want to kill myself too. So the two of us, we should die. Now asemfa na asem enko. So the neutrophils kill microbes by free radical, that's a hydrogen peroxide, superoxide radical, or hypochlorite, or nitrite radical, or by cellular suicide. Okay, so interferons. Interferons are also produced by viral they are chemicals produced by viral infected cells to interfere with viral replication. So when a virus or a cell has been affected or infected by a virus, it produces interferons, and the interferons will now kill the viruses, because any healthy cell that has been attacked by viruses tries to defend itself by producing chemicals against the virus, and these chemicals are called interferons. So we have interferon alpha, interferon beta, interferon gamma, and the alpha interferon is produced by B cells, monocytes, and macrophage. Beta interferon is produced by fibroblasts and viral infected cells. Then gamma interferon is produced by activated T cells and natural killer cells. So whenever you hear fibroblast is for what? Wound healing. It is type 3 collagen. And it's type 3 collagen. It comprises of type 3 collagen. Collagen type 3. Type 3. With this one mediates wound healing. It mediates wounds when you have a sore. Wound healing. Collagen type 3. Wound healing, whenever you see fibroblasts, they are talking about what? Wound healing. That's what causes wounds to heal. Okay, so let's continue. So the interferons, they are chemicals produced by viral infected cells. So those who are having hepatitis B, hepatitis C, interferons are very expensive. Some of them are even selling them for 70 million. Some are selling them 15 million. Some are selling them 100 million. They are very expensive and very, very effective. So we have complement system. Pay very particular attention to this section. But before I start the complement system, let's take a break.